The optimum pH for an enzyme is the pH at which enzyme activity is highest. Different enzymes have a different optimum pH. This diagram shows a pH scale that goes from 0 up to 14. A pH of around 7 is neutral. A pH value of around 0 is very acidic, and a pH value of around 14 is very alkaline. These two enzymes have been placed on the pH scale around their optimum pH. One of these enzymes sits around the optimum pH for working in the human stomach, and the other sits around the optimum pH for working in the intestines. Before going on to the next slide, have a think about which one is which. Enzymes in the human stomach work best at an acidic pH. Enzymes in the small intestine work best at an alkaline pH. Therefore, this enzyme right here would be the stomach enzyme, and this one would be the intestinal enzyme. Remember that the optimum pH for these enzymes is where the largest number of successful collisions take place between the enzyme's active site and the substrate molecules. So what would happen then if we were to move away from the optimum pH? Any change away from the optimum pH lowers enzyme activity. Let's look at a graph showing a typical example of enzyme activity at different pH values. So on this graph, we're going to plot pH on the x-axis and enzyme activity or rate of reaction on the y-axis. The curve would look something like this, with the bell peaking at around pH 8. This indicates the optimum pH for this enzyme. You can see that any movement away from the optimum pH causes decreasing activity of the enzyme. Make sure that you familiarise yourself with graphs like this. It's important that you can relate enzyme activity to pH changes for your exam. Now, let's have a look at why the enzyme activity goes to 0 at these extremes of pH. Enzymes are denatured by extreme pH conditions. Remember, when denatured, the shape of the active site is altered so that the enzyme can no longer bind to its substrate. On the left-hand side here, you can see a normal enzyme. The active site still fits the shape of the substrate. However, if we were to expose this normal enzyme to very high or very low pHs, we would end up with a denatured enzyme. The very acidic or alkaline conditions damage the enzyme structure, breaking bonds within it, which means that the enzyme can no longer bind to the substrate. The key no longer fits the lock. This change in the enzyme shape is permanent. It cannot be reversed.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy-to-follow videos, and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE Biology course. See you there!